Hello everyone, my name is Abby and I'm going to be walking you through the Yeast ORFan Gene Project Lab Module 6. In this module, we will use tools to help you predict where your gene's product is most likely to be found in the cell based on its sequence patterns. These tools include Phillius, TargetP, NucPred, and the Yeast Localization Database. So let's get started. So we're going to get started by opening up the guide and the worksheet from the Yeast ORFan Gene Project.com. And we're going to get started by using the Phillius database first. So I'm just going to open up that website. And you are also going to need to go to the SGD and get your FASTA sequence. I am going to be using TMN2 and downloading the protein sequence. And make sure you keep that up for the rest of the video because you'll need it. So we're going to paste it in and click Run Phillius. And that's what it'll look like when it loads. And you might not get any results for this, and that's fine. So we need the predicted protein type. And mine is a transmembrane with signal peptides. And then the type confidence is 0.58. And the topology confidence is 0.56. And then I'm going to take a screenshot of the prediction image map and a screenshot of the prediction sequence map and place those into their corresponding sections in the worksheet. Alright, and going into what this means, so this is fairly similar to the hidden Markov model. It's showing that we start as non-cytoplasmic for my protein. It's green, it says non-cytoplasmic. It also shows the type confidence and what residues are involved in that non-cytoplasmic segment. And then the yellow is showing when it's in the transmembrane, and it shows it 9 times, similar to my hidden Markov model that showed it crossed 9 times. And then this just lays out where in the sequence is it the transmembrane helix, non-cytoplasmic, cytoplasmic, and a signal peptide. So now we're going to copy down the segment types based on the prediction sequence. So my first is a signal peptide. The type confidence is 0.56. And the residues are amino acids 1 through 18. So then the next segment is non-cytoplasmic. The type confidence is 0.98. And the residues are 19 to 309. The next segment type that comes up is the transmembrane helix. The type confidence is 0.90. And the residues are 310 to 332. And then for the next segment, we have cytoplasmic. The type confidence is 0.94. And the residues are 333 to 375. And then I would ask for your professor, mine has a lot more after that, so I would ask your professor if they want you to get the rest of it and record the segments after that in their corresponding like transmembrane, non-cytoplasmic, cytoplasmic, or signal peptide. And they all have different type confidences as well. And now onto TargetP, so I'm going to open that. And we're going to be clicking on server 1.1 for the TargetP. And you can use TargetP 2.0 and see what results you get, but this worksheet is specific to 1.1. So I'm going to copy my FAFSA sequence. I'm using MGE1. We want non-plant selected and no cutoffs, winner takes all, and then click submit. And this is what it looks like when it's loaded. And we want the MTP score, and mine is 0.918, and the SP score, which is 0.008 for me. So the MTP and SP are reported as probabilities out of 1 that your protein contains a mitochondrial targeting peptide, or the MTP score, or a secretory pathway signal peptide, or the SP score. Then we also have RC, or the reliability class, which is from 1 to 5, and 1 indicates the strongest prediction, and it's a measure of size of the difference between the highest or the winning output and the second highest output scores, and mine is out of 2. So then, do you believe your protein contains a mitochondrial targeting peptide or a secretory pathway signal peptide? So my MTP score is 0.918, so I believe that my protein contains a mitochondrial targeting peptide, as it has a score close to 1 and not a secretory pathway signal peptide, as it has a lower score. Alright, and now moving on to NucPred, I'm going to open the website and get my FAFSA sequence. And we want to click on the single protein hyperlink and paste our FAFSA sequence in there and click submit. And this is what it'll look like when it loads. And you're going to want to take a screenshot of that. So, and then that down there explains the NucPred score, and mine is 0.04, which is showing that it's not nuclear. And if you look at the sequence, most of mine is in the blue-green, so non-nuclear. So this is saying if it's 0.10, the fraction of proteins predicted to be nuclear that are actually nuclear would be 45%, and then the traction of the nuclear proteins that are predicted, or the coverage, is 88%, and that changes with the different NucPred score. So mine is 0.04, so it's lower and it's not likely nuclear, and you can also see that from the coloring of the sequence. So no, it does not likely contain any nuclear localizations sequences. And again, most of mine is in this blue-green. If it was more so in this yellow to the red, it would more than likely have nuclear localization sequences. So now onto the Yeast Localization Database. So I'm going to open that up. And for this one, I am going to be using the gene YOL019W, because for both TMN2 and MEC1 there were no results. So the initial shows cell periphery and vacuole, and then the final also shows cell periphery and vacuole. And then those are the fluorescent protein tagging images. So mine says technical problem, so that should typically be a number. And then these are the fluorescent tagging images from the GFP that you're going to want to take a screenshot of. And paste into the worksheet. So based on the GFP protein fusion, it looks like it's vacuolar, or I believe cell periphery. So, and then summarizing the data from the last two modules, you're going to want to look at all of the hypothetical stuff from like p-sort2 and your hidden Markov model, and then all of the information from this module, and see what lines up, see what's reoccurring, and comment on that. I really cannot because I used example proteins for different modules before, and for the localization database, I also did not use TMN2, so I can't really comment altogether. So you would just want to take into consideration what is reoccurring within all of the databases, what does it look like it could strongly localize to, and then make a hypothesis based off of that information from this module and the last module. Thanks for watching. I hope that this video was helpful. For more information, go to the yeastorfanproject.com, and this video was funded by the National Science Foundation. Thanks!